Hello and welcome to BioRules video lessons. Now we are going to be getting to our final class about how impulses are transmitted along a nerve cell. Actually now it's not along a nerve cell, now we're going to see how the nerve impulse that was transmitted along a nerve cell is going to be transmitted to the next nerve cell, which is called a synapsis. So today's class is going to be about synapses, which ultimately is about the connection between nerve cells or between the nerve cells and the effectors, the ones that are going to be making things to work. So what we have here are connections between nerve cells. This here is what's called a synapsis, and we are going to be happening there in the end of an axon, which is this guy over here. This is the end of an axon, and the beginning of the next nerve cell, which is a dendrite, which is going over there. So this transmission here is called a synaptic transmission that happens in the synapsis, which is the connection between those two guys. So we have here another one. You have an actual potential traveling around, and when it reaches the end, we are going to be having the signal going. So basically, you already know about the actual potential, the saltatory impulse, all that we saw over there. We have an actual potential traveling just like that, what we saw in the previous class, and the actual potential goes making things becoming from negative to positive to negative back again, and when it reaches the end of the axon, when it reaches the synaptic knob, the presynaptic knob, the one that's before the synapsis, this one over here, we are going to be having, it becomes slightly positive. So it will get over there, and let's see here, let me finish my little drawing. We are going to be having this guys here acting. We have calcium channels, we have vesicles that are going to have neurotransmitters, and we're going to have the actual potential. So when the actual potential gets in there, see what just happened? The actual potential opened the calcium. So because the threshold for the calcium now is reached, when the calcium channel, dependent on voltage, is open, calcium moves in. Yay, calcium moves in. And calcium is an interesting ion. Calcium can be used for your teeth, calcium can be used in your bones, but calcium is also used by motor proteins to move. Your muscles, they move on calcium. Calcium signals the nerve cells to move, the muscle cells, sorry, to move. And they also signal motor proteins in the membrane that carry vesicles around to start moving. So when the calcium gets over there, the calcium vesicles, they move toward the membrane. They are carried by those little guys that carry stuff inside of the cell. They carry it in the direction of the membrane of the synaptic knob. And when this vesicle travels, it gets into the membrane, it touches, you have exocytosis of neurotransmitters, and the neurotransmitters being released, they jump, and now they are available to the next nerve cell, so they act on the next one. This here is what just happened in the presynaptic knob. Okay? Then I'll put everything here back again. You see it happening quite fast. To illustrate to you what's going to be happening in the postsynaptic knob. The postsynaptic knob, you have the same stuff that you had before. We have sodium outside, you have potassium inside, sodium-potassium pump, resting potential, all that stuff. But the difference is that in this knob, we don't have voltage-dependent sodium gates. We have neurotransmitter-dependent sodium gates. So these guys over here, they are not waiting now for an electrical signal. They are waiting for a chemical signal. When we have there the impulse, the calcium channels open, calcium moves in, calcium is going to be signaling for the membrane, the vesicle travel the neurotransmitter to the membrane, the exocytosis, and when you have the exocytosis, the neurotransmitter goes there and boom, touches the little place there in the gate, the sodium channel gate, and when it touches over there, look what's going to be happening, open. So what happens here is that neurotransmitters, they open, in this example, sodium channels. And when they open the sodium channels, sodium enters. And when sodium enters, it makes things become slightly positive. If it gets enough positive, reaching the threshold, the minus 55, then you have an actual potential. And then actual potential here is going to be traveling along all the way through the axon to send a signal to another one, and then to another one, and then to another one. So what you just had here was an electrical signal, electrochemical signal, becoming a chemical signal that becomes electrochemical signal back again. The nerve impulses are transmitted along nerve cells by electrochemical impulses, but they are transferred between nerve cells in the synapses by chemistry. It's neurotransmitters. In this example that are open in the sodium channels, we are talking about acetylcholine. That's one neurotransmitter. There are other neurotransmitters like norepinephrine, which is different from the adrenaline or epinephrine. Noradrenaline or norepinephrine is the neurotransmitter. Epinephrine or adrenaline is the hormone. But they do more or less the same idea. They are going to end up having similar effects. That's why they have the similar name. There are other neurotransmitters like serotonin, dopamine, and so on. So these neurotransmitters, each neurotransmitter has a certain effect. But they are going to be playing with channels in the postsynaptic neuron. Okay, so that's how it's going to be happening. So here you have a simplification for you. You have calcium entering. Calcium entering is going to be making the vesicle to move, release neurotransmitter. Neurotransmitters touch here and start a new actual potential. Okay. The beauty here is that when you have a signal going in there, now it goes there. You have an actual potential, gets to the end, opens the calcium channels that put some calcium in, and then is going to be making some neurotransmitters to be released, and these some neurotransmitters are going to be opening some sodium channels, but if it's not enough to reach threshold, there is no signal. And then comes the question, okay, how do we get a signal? For a signal to be passed on, the signal needs to be strong enough. Quotation marks now when this is strong, because actually it's not strong. It needs to be strong in the sense that it needs to signal, let's open this stuff. Like in the arc reflex that I showed up a few classes before, when you have a strong stimulus in your skin, it might signal for the relay neuron to trigger the motor neuron to make you move. And if the signal, the pain is not strong enough, it doesn't send that signal. So what is to be strong enough? Definitively is not going to be a bigger actual potential, because you don't have bigger actual potential, because actual potentials, they go from minus 70 to plus 30 or 40, something like that, to minus 70 again. So there's no variation here, so you cannot have a bigger actual potential. So how can we release more sodium inside of the postsynaptic neuron to be able to reach threshold and start a new signal? Simple. We need to release more calcium. So more calcium move more neurotransmitters, and more neurotransmitters open more sodium channels. That's going to be triggering enough to reach the threshold. So how do we do that? Quite simple. Instead of sending one big one, what can be done is that more actual potentials can be sent one just after the other. When I say just after the other, I'm not saying 1 or 2 milliseconds, because 1 or 2 milliseconds, the regions in the axons, they are still in the refractory period, but no, 4 or 5 milliseconds later, 3 or 4 or 5 milliseconds later, you can send another one. So if you send an actual potential, 3 milliseconds later, you send another one, 3 milliseconds later, send another one, you just reached the end, so you put more calcium is being opened, so you have more neurotransmitters being transported that are going to be making more sodium channels to be open to reach threshold, and when reach threshold, it triggers a new actual potential that signal ahead. So this is one of the ways that you can modulate, that your synapses actually can decide, are we going to be having a signal here or not? Okay. In class, I'm going to show you other things, because neurotransmitters, they are not only going to be excitatory, they can be also inhibitory. So I'm going to be using two colors here, red and blue. So let's say that the red here opens sodium channels. What happens if you open sodium channels? I'll make a little graph here on the corner. Let's make a little stuff over here, and I'll make this in, let's change color. Let's make this in brown. And so what happens here? I have minus 70, okay? And then if I release a little bit of sodium in here, this guy becomes slightly positive. It went over there, but not enough to reach threshold. And then this guy releases also, sends a signal, releases a little bit of sodium in there, and man, look, it's almost in there. But this other one over here releases actually neurotransmitters that open potassium channels. If they open potassium channels and potassium then leaves over here, oh man, this became a little bit less. So let's make it less over there. And then this guy here releases sodium as well. So sodium goes over there, and neurotransmitters that are going to be stimulating the synapse, so it's opening sodium channel, so it releases sodium, it gets a little bit more positive. And then this other one here, oh yeah, sodium again. And then, boom, voila, reach threshold, and you have a signal, boom, you have a signal traveling over there. So up to 10,000 nerve cells can connect to one cell body on one nerve cell to modulate. Are we going to be responding to this? Yes, no, no, yes. So you have a stimulation, you have not stimulating, and so on. So the synapses are actually deciding, are we sending a signal ahead or not? And it all depends on who is sending a signal to where. Okay, remembering that each nerve cell makes up to 10,000 connections, and you have trillions of synapses in your brain. So the brain is quite complex, but the idea is simple as this. Okay? Take your time now to do a little quiz, and I'll see you in class. Definitely we are going to have a lot to talk about. Bye-bye.